Today I'll be discussing Chapter 4, Cellular Metabolism. My objective is 4.3, describe how enzymes control metabolic reactions. To start off, just to define an enzyme, they are complex molecules, and I believe most of them are globular proteins that function to lower activation energy in cells. By doing this, enzymes are enabling reactions to proceed faster, and because of this, enzymes are called catalysts. The chemical reactants to which an enzyme binds are called the enzyme substrates. There may be one or more substances just depending on the particular chemical reaction. In some reactions, a single reactant substrate is broken down into multiple products. However, in others, two substrates may come together to form a larger molecule. In the figure depicted right there, we can see that the substrate would be the green figure and the enzyme is the yellow. They do bind at the active site, and each enzyme is specific to a substrate. They recognize that specific shape of the substrate, which is the one that they react to. There's a lock and key model right there. For example, in an anabolic reaction, small molecules are built up into larger ones, and this process does require energy. Again, in this picture, we can see that the green and the circle substances come together, they bind with the enzyme, and the enzyme is able to make one large molecule from the two that they started with. In contrast, however, for example, in catabolism, reactions are large molecules that are broken down into smaller ones. This process does release energy. Again, in the picture, you can see that the enzyme starts with binding to a large molecule and they bond, and you end up with two different products, smaller molecules than you started with. Enzymes control the rates of both catabolic and anabolic reactions, but of course, in order for metabolic reactions to occur, energy is needed, and most of the reactions critical to a living cell happen too slowly at normal temperatures to be any use of the cell anyway. So this process does require ATP. In cells, enzymes initiate oxidation by lowering activation energy. Oxidation releases energy from glucose, which is then used to power cellular metabolism. Enzymes do this by binding to the reactant molecules and holding them in such a way as to make a chemical bond, whether it's by breaking or forming, as I mentioned before, and these processes are able to take place more easily. These processes can be reversed, and the enzyme does not, it can be reused because it doesn't get consumed in the reactions. And here are my sources.